Okay, perfect. So 6:03. I guess we should make a start. I just want to check that we're recording before we begin.
Yeah, all good, mate.
Cool, perfect. Okay, so thank you everybody for joining us today for our journal club. I'm very honored to say that we have Professor Watts today. Who's going to be leading our journal club this evening. So just giving everyone a brief history on Professor Watts. He previously was at Cambridge, but now is a professor at Birmingham and is very heavily involved with the newly established brain cancer program there. Due to his keen interest in helping out with the student body, we are really honored to have him here today to discuss the Tessa Jowell BRAIN MATRIX trial, in which he is the chief investigator. It's a very unique trial, and I'm sure people are going to learn a lot about it today, and it's had a very, very large grant of, if I'm not mistaken, 2.8 million, so it should be very interesting to hear today, yeah. But yeah, without further ado, I do welcome Professor Colin Watts.
That's fantastic. Thank you, Joshua. What I'm going to do today is just put into context where we are with the brain matrix trial, the background that led up to it in terms of how we think about brain cancer and translating that into clinical trials and so on. Brain cancer is something that is not it's not something that's commonly talked about, certainly at undergraduate level, and only if you specialize at postgraduate level does it become anything like something that people talk about. It's not on a medical oncology curriculum. It's an optional extra, but in actual fact, it's a case of unmet need because it has a significant amount of morbidity and mortality. It accounts for 2% of cancers, but it represents 7% of cancer deaths. And this is because you might be more likely to get prostate cancer or lung cancer as a middle-aged man or breast cancer as a woman. But if you get those tumors, there is often something that can be done. And it's important to recognize that if you get brain cancer, it is often very difficult. And brain cancer is one of the it's the commonest solid tumor in children. If you then look at what happens when patients enter into a specialist care pathway, about 2/3 of them will have some sort of surgical intervention with the objective of trying to get a tissue diagnosis. About 1/3 of patients will have no treatment at all because the disease is too advanced when they present. So there's clearly a need to get earlier diagnosis and earlier detection. Brain cancer, the WHO describes about 130 different types of brain cancer. And the good news is that the common ones are astrocytomas and oligodendrogliomas. And these in turn can be broken down in terms of grading, WHO grading. And that grading is basically built around what these things look like down a microscope. And one of the things that we want to do in brain matrix and more generally is bring molecular data into understanding classification in the context of real world problems. So surgical management, essentially the priority is of establishing a diagnosis, but it's also really important to remove as much of the disease as we can. Unlike other cancers, such as breast cancer or colon cancer, it's not an option to remove the disease plus a bit of normal tissue around it. So we have to be much more careful about how we operate on these patients and how we treat them once we've operated on them. Therefore, removing disease, removing the volume of disease can actually help radiotherapy and chemotherapy, because as those treatments start to work, they cause cell death. That causes bystander effects, and you end up with swelling. And if you've not debulked it, then sometimes the symptoms, the neurological problems associated with swelling can prevent the oncologists from treating the patients as aggressively as they might like. Lastly, I would also argue that it's important to collect tissue for research, because this is one of the least understood cancers, despite being extensively sequenced. We still don't know a lot about it. So overriding all of that is it's important to keep the patient safe, because if we cause an additional neurological problem, as this data here shows, if we cause a new post-operative deficit, whether it's language deficit or motor deficit, then you can significantly impact on survival. So it's really important that we don't do any additional neurological disease. To do this, we develop a toolbox, which involves using navigation techniques, awake surgery, intraoperative monitoring, both awake and asleep, the fluorescence-guided surgery, for a privileged few, intraoperative MRI, although for high-grade adult cancers, it's probably not as useful as it might be, for example, in pediatric tumors, and intraoperative ultrasound. In essence, very few units use everything, but the idea is to develop a multimodal approach to surgical management. And that involves integrating the technologies you have, whether that's the neuronavigation systems that allows you to track the position of the tumor in space relative to a fixed point, in that case, the arc here. Integrating that with your operating microscope, effectively turning your microscope into a giant pointer, and then using mapping during surgery, both cortical, as you see here, and also subcortical, to identify functional areas and avoid that. If you take examples such as this one, where we have disease in the left temporal lobe affecting speech and language, and we can integrate doing awake surgery with the use of fluorescence, which allows us to distinguish what looks like normal tissue under white light conditions, clearly abnormal under fluorescent guided conditions. It also, using this technique, also allows us to get to a point where we can objectively say, we've removed this fluorescent disease, it's time to stop. And then when you do the post-op scans, you find that the disease, the contrast enhancing disease, has been removed. Patient is well and can go home. While we're doing everything we can to improve the neurosurgical work and the oncology work, it's also important to recognize that there's a lot of work going on to understand how these tumors grow. One of the earliest events is mutation of the IDH gene. And what that does is essentially marks the distinction between the very aggressive IDH wild type high-grade gliomas, usually glioblastoma, and the IDH mutated, which then undergo further stratification based on whether or not they got 1p19q co-deletion. So loss of 1p and 19q identifies patients who have oligodendrogliomas. If it's absent, these are astrocytomas that are less aggressive than the IDH wild type tumors. And so it's important to understand that, because it doesn't matter how good our treatments are, we need to understand the results and prognosis in that context. So if you look at, for example, this graph at the top here, which looks at grade 2 and 3 tumors, you have tumors that can be defined on the basis of TERT promoter mutation and IDH mutation. You have triple positive, which are TERT promoter mutated, IDH mutated, and 1p19q co-deleted. And these form a group of tumors where the median survival is well in excess of 10 years. You have other tumors such as these, which are just IDH mutated, which are slowly progressive. And then you have these here, which are identified just by the TERT mutation, which are very aggressive indeed, but actually look like less aggressive tumors down a microscope. And these are biological glioblastoma, these grade 4 lesions here. And it's important to identify these as quickly as possible, because the treatment for these is very different to the treatment for these. It's also important to understand that these patients with a much more benign pathway prognosis, we need to be very careful not to give them any additional neurological deficits that they don't have to live with for many years. So we can begin to stratify these patients into different groups. But nevertheless, you can still have a patient who is diagnosed with a GBM who may well be dead within a few months. But then you get other patients with a diagnosis of a GBM who are still alive at 5 years down the line. There's clearly a lot we don't know in terms of stratifying these patients based on what we see in the clinic. So on the basis of all this, it's possible to start thinking about how we might tailor surgery, how we might refine what we do, and balance risk and benefit in different ways. De-escalating tumors if they're potentially chemosensitive, such as the oligodendrogliomas, but still trying to remove as much as is safely possible. Improving maximal resection for those that are relentlessly progressive, where the balance favors being a bit more aggressive and going back in if there's residual disease. And then these most aggressive tumors where we need to get clearance as quickly and efficiently as possible. Having said all that, what do we do in terms of treatment? When you have these IDH mutant or IDH wild type 1p19q intact versus co-deleted, or another group I've not mentioned, the H3K27, you can start to stratify all these. But at the end of the day, if you look at this chart for treatment, one of the saddest things is that at the end of the day, we're looking at radiotherapy, temozolomide, and PCV chemotherapy. Only 3 different modalities. There's an urgent need to find new drugs. And it's not for want of trying. You look at this picture, all of these drugs here have been tried in patients. They've all had sufficient preclinical mechanistic interrogation to justify a mechanistically based hypothesis. They've gone into early phase clinical trials, and all of them have failed. And there have been many more added since this slide was put together in 2013. So in essence, we need to think about doing things in a different way. Why is all this happening? Well, one of the reasons is this issue around complexity. The idea that scientific methodology tells us to develop experimental model systems where you change one variable at a time. And in clinical trials, that translates as something like standard of care versus standard of care plus X, where X is your new treatment. And for cancer, particularly for complex cancer, this really needs to change. We know that there are dynamic interrelationships between different aspects, such as the tumor growth, the tumor microenvironment, its immune activity, the immune environment, the metabolic environment, and so on. And under these circumstances, recognizing these dynamic interrelationships allows us to think about treatments that are synergistic and to be used in combinations in order to tailor them to more individual or groups of patients. The other thing about complexity is how can we understand, how can we construct a framework to think about it? And one of the things is to apply Darwinian evolutionary approaches to think about the tumor as arising from a population of cells that are under selective pressure. And then as you treat them, whether you operate on them, give them radiotherapy or chemotherapy, you create a different selective ecosystem that allows ultimately the emergence of resistant disease as treatments fail. How can we interrogate this, and what role do surgeons have? Well, one of the challenges is recognizing that the tumor contains lots of different clones, lots of populations of cells, all fighting for resources, a bit like all the animals alive on the earth today. They're all fighting for oxygen, for space to grow, for food, and so on. In a tumor, depending on where you sample, you might get one population and think that that's it. The tumor is comprised just of that population. You might take a sample elsewhere and find you get 2 or 3 different populations, and you make a very different conclusion about what is going on in that particular tumor of that patient. We have to think about not just that, but understanding how we can interrogate the tumor and sample the edge of the tumor. And as I've already alluded to in brain cancer, this is particularly challenging because of the functional issues around the function of the brain and the fact that this is a cancer growing in a functional environment. If you have white light and you're operating, you don't know if you've still got disease or if you've removed it. And therefore, it's very difficult to repeatedly and objectively sample different parts of the tumor. If you're using fluorescent resection, you know that you've got to the edge of this high-grade cancer, then you can say reliably that you can take these samples as margin samples and understand what it is that you're leaving behind. Because obviously, if we take away the mass, we still leave disease behind. And it's that disease which leads to the recurrence and progression. And you can integrate all these techniques. You can use the fluorescence resection, all the intraoperative mapping, the neuronavigation, and you can develop techniques to spatially sample these tumors, as we've done in the past, and look at it and use those samples to identify clones that have probably been there since the earliest stages of that tumor. And so we can look at this and argue that there are distinct types of tumor, distinct subclones of cells within each tumor fragment, and that these work in complex hierarchies. And it's the genetic diversity of all these clones, all these different species of animals within the environment, fighting and growing, that are probably the basis for selection of an emergence of resistant disease. More recently, work on glioblastoma has taken single cell sequencing, recognized atlases of genetic sequencing, and coupled that with experimental models to identify the fact that there are 4 cell states driving malignant cells, and that these cell states, it's the genetics and the microenvironment that influence the frequency of cells in these states, and that we can look at this in terms of subtypes that we see in the clinic. Putative hierarchies can exist for some types of glioma, such as the IDH mutant glioma. You remember that relentlessly progressive one, or the H3K27, where there are clear populations that give rise to other populations of cells that stop proliferating, differentiate, and contribute to the tumor volume. Glioblastoma seems to be very different. It seems to be designed around cells that transition between different states. And so this transition between states reflects the idea that there is a synergy between what we call subclonal evolution and cancer stem cells. In other words, these cells can act as units of evolution and units of selection within a population. And if you deplete one particular subclone or subpopulation, then other subpopulations can evolve and adapt to repopulate the whole mass of growing tumor. So switching between these different states, our ability to identify and define these states using stem cell markers, oligodendrocyte precursors, astrocyte precursor type markers, and so on, allows us to begin the process of trying to interrogate how this might change. So how does all of this influence how we think about clinical research and clinical trials? Because, as I've shown you, the standard model, the ones that work perfectly well in other contexts, aren't working here and haven't worked. One of the problems is that our current practice doesn't adapt and recognize the fact that the disease evolves. And as the disease evolves, that evolution manifests as a more aggressive disease phenotype. So what happens normally is a patient will present to a specialist multidisciplinary team. They'll be reviewed. They'll undergo some sort of diagnostic surgery, whether it's a biopsy or a craniotomy. You'll get some fairly crude markers that will give us some prognostic information. They'll then have some treatment, and if they're lucky, they'll go into a period of stable disease. But inevitably, there's going to be disease progression. And after that, it's best guess treatment, because we don't have any further biological data and we don't understand how the disease is changing. We need to evolve how we do it. We need to get that first surgery to deliver a genomic-based diagnosis that copes with the whole genome and increasingly probably the epigenetics that are going on. We need to develop techniques that will allow us to minimally invasively monitor any transitions, and that includes blood and CSF and urine as liquid biopsies, and possibly imaging to monitor changes. And then we need to think about how we can interrogate that tumor again, whether it's through second surgery or cell-free DNA or other alternatives, so that we can evolve the diagnosis that we have and refine it, and in that way allow better treatment to adapt. So when you think about this, you can come up with these little figures like this one, where we're looking at treatment A versus treatment B versus controls in a mixed population. You then identify certain patients that respond very well to treatment B and certain patients that don't respond very well to a given treatment, such as treatment A. And you can then stop one arm and expand and enrich for the responder phenotypes to develop the trial further in treatment B against a matched control population. And these adaptive trials will eventually start to change and replace our traditional approaches. But to make this work, we need to have a range of drugs available to try. Otherwise, we have no treatments. We need to understand how we can get real-time response data, and that's critical, because we need to know as quickly as possible if a treatment is failing or not working. We need to manage the data. Data management is actually a real challenge. Taking data outside of an NHS trust firewall, manipulating it, then bringing it back across the trust firewall, de-anonymizing it, and then taking that back to the treating physician, the oncologist and the patient themselves. We need better outcome measures, and we need national and international collaboration. And that is what Brain Matrix is all about. This is what this slide, which looks nice and even a neurosurgeon can understand it, this is what it looks like in real life. These are extremely complicated, challenging trials. It's not meant to, you're not meant to read all this. It's just meant to illustrate the simple fact that when you try and translate into the real world, these complex trials become very challenging. We need to understand the impacts of the microenvironment that the tumor is growing in. Its biology, how does it feed? Where's it getting its nutrition? What is the tumor metabolism? How is it defending itself? How is it manipulating the immune environment? And how is it growing? We need to develop better preclinical models so they more accurately represent what we see in patients in the real life. We need to understand how different components of the tumor interact, whether that's extracellular matrix, tumor macrophages, microglia, T cells, and then of course the vascular endothelial cells and the blood-brain barrier. And we need to think about in silico alternatives to bring together multiple dimensions of modeling in order to better improve and understand and quickly evaluate therapeutic options. And of course, the trendy thing at the moment is about patient avatars, and that involves linking clinical trial data and being able to interrogate the tumor in much greater detail from each patient. If we develop a precision medicine approach, we need to move beyond simple morphology, what something looks like down a microscope and how you stain it with an antibody. We need to be much more detailed in our characterization. And that's already started with large-scale methylome subtyping of brain tumors. And this proof of concept has been established. And actually, in fact, it changes the diagnosis in about 12% of patients. And that's absolutely critical, because you can't develop precision therapeutics if your diagnosis is not as accurate as we can possibly make it. So if we're looking at a precision medicine approach in the future, it's going to use artificial intelligence and machine learning. It's going to integrate multiple types of data, and it's going to do so throughout the patient journey. It's going to bring together data sets coupled with quantitative imaging. At the moment, most of our imaging is qualitative, and we need to understand and select, as I've said, treatment response to better identify treatment failure. And we need to understand the range of clinical phenotypes that you see in real life, as I've said. So every patient needs to be able to be interrogated over the course of their therapeutic lifetime. So that's the background, and that's the sort of theoretical basis of what we're trying to do here. And moving to Birmingham was and has enabled me to start realizing and begin the journey to realize that vision. Because in order to do it, it's a question of integrated thinking. And it begins by understanding the patient's pathway of care and then building around that an architecture that essentially makes the clinic, the operating room, the laboratory, a synergistic whole, a black box, if you like, that will churn through and engineer results, taking tissue, analyzing that tissue, identifying drugs, bringing them back in to check that the drugs penetrate the tumor, ensuring they deliver on the mechanism they're supposed to do. But it's much more than that. It's about improving the preclinical models that we can do and developing liquid biopsy technologies. It's about improving our diagnostics, as I've said, and collecting patient data and enabling drug development. In other words, bringing the lab to the patient, making the operating room an extension of the laboratory and the clinic an extension of the laboratory. In doing that, we will create an adaptive learning environment for the whole of the patient journey that begins at presentation to the tumor multidisciplinary meeting, go through initial genomic diagnosis, treatment, stable disease, progression, and refine that diagnosis as disease progresses, and ultimately even in an effort to understand how the disease finally wins, to look at what the disease looks like in patients who've finally succumbed. We need to capture the clinical phenotypic diversity that I've mentioned. We need to look at all types of glioma, not just one type. And we need to look at multiple types of data, how we curate it, how we use it, how we deal with the ethics of anonymizing data so it can be manipulated and analyzed anonymously, and then identifying it once we get back across firewalls so it can be used by clinicians and by patients. We need to follow up for life, as I've said, and we need to set standards of excellence for engagement. So that then is where we're going with the Tessa Jowell BRAIN MATRIX. It's a national effort that involves a clinical network of 10 centers at the moment, and we're actively seeking to begin to expand that. It's bringing in state-of-the-art diagnostics using methylation-based arrays that I've mentioned, and it's integrating that with genomic data and doing so in an NHS routine clinical practice. And it's then taking all of that information and utilizing it to deliver new therapeutic arms through an early phase trial structure. So what does that look like? What we're aiming to do initially, first step, is create a platform that will allow all this. It's an enabling platform. And as I've said, it consists of clinical hubs. We have 10 in the first phase, coupled to an imaging hub and a molecular hub. And the idea is that information, imaging data, biological data, and clinical data are collected, integrated, and then fed back as aggregated data, both to data repositories where the data can be stored and made available anonymized to researchers anywhere in the world, and also identifiable data that can be used within the context of treatment and getting clinical study arms going. In practice, if we look across 10 centers and reckon, based on survey work that we've done, you get about 100 patients with a glioma per year. If we make the entry criteria really simple, any surgical patient can be eligible, then we have the capacity to recruit lots and lots of patients. We can collect that tissue, we can do whole genome sequencing, methylation, we develop tissue and imaging banking, and we develop a centralized repository of integrated data and make that available to scientists and clinical researchers around the world. We provide what some people have referred to as a continuous value chain. In other words, we develop clinical study arms. We develop clinical study arms that are directed against specific and identifiable mutations or mechanisms that have been identified through the sequencing and through the diagnostics that we do. The reality is we need to manage expectation, because the number of patients going into this arm is actually probably going to be fairly small, certainly in the first few waves of evolution, until we get a lot more drugs available. And if that is the case and there's no actionable mechanism, then patients can go into prospective observational data studies, to generate data that can be used to inform further treatments, inform clinical policy based on what's available. If patients do have actionable genetic mutations, then either a drug's going to be available or not. And again, if it's not, they can go into this backbone, and we can flag the fact that we could do with drugs targeting certain specific mechanisms. And one of the areas where this will expand in the near future is around repurposing existing drugs. If there are novel drugs are available, then the next question we want to ask is, do these drugs get into the tumor in the concentrations that we would expect based on the preclinical data? And does it do what it's supposed to do? In other words, what's the PK and PD, the pharmacokinetics and pharmacodynamics? We can do alternatives if we don't know the answer to these questions. For example, using the window study approach, we can give a drug, then interrogate and operate on the patient, take tumor samples, and analyze those tumors to see if there is drug penetration, if the mechanism is working, or if we can move straight on. If we already know the drugs get into patients, then we can move rapidly into early phase study arms, multiple study arms, with the idea of saying, okay, let's set a significance limit that is higher than we do at the moment. Because as I say, a significant number of drugs seem to have some promise of effect at phase 2, but then bomb and don't work at phase 3. So it's about creating this environment, this hierarchical value chain, if you like, this way of doing it that will allow us to progress and process patients. So that moving forward, the next generation of clinical trials will support a research strategy that promotes interdisciplinary action. So we have a pathway of care, we have a trial protocol, and we're recruiting patients because the questions that we're asking are relatively straightforward. Can we get an integrated diagnosis back to a patient within 28 working days? At the moment, it takes a bit longer than that. But we can do a lot more going forward. We can take patient samples, we can learn how to do this safely using advanced biopsy techniques and liquid biopsy. We can then feed that into a research pipeline that will generate targets, drugs, diagnostic technologies, new surgical technologies, and so forth, that will allow us to create a virtuous circle of adapting treatments and developing ways of doing new trials and getting drugs to patients faster. And there's been a lot of talk about this, because one of the things that we've learned about from the COVID work is that if you embed this kind of research into routine clinical care pathways, and you adapt and accelerate the techniques to overcome what are currently perceived as being barriers to work. An example would be sharing data between trusts and hospitals and different departments. Then we can really make a difference, and that's one of the lessons that we've learned from how things have been done in the COVID pandemic. So there's a lot going on. That's the essence of what we're trying to do with Brain Matrix. It's completely novel. There's nothing like this precisely going on anywhere else in the world. And one of the advantages we have is that we have a healthcare system that is defined, single payer, single user, which means that in theory, at least, most patients, wherever they're treated, will get the same standard, the same care that we can offer. And that creates a fantastic research environment to develop clinical research and clinical trials. This is my last slide. I didn't want to spend a long time talking. I wanted to provoke some thought. And this is something from Bill Gates. At the end of the day, if you're the medical students and junior doctors of now, then you guys are going to be the ones doing this in whatever shape or form it will evolve into going forward. How we can integrate, interact, and work with the young doctors of tomorrow in Brain Matrix today is something that is open for discussion. And let's see how we can all work together and get engagement, so we can not only do all the trials that we want to do the research that we want to do, but we can also train the future clinical trialists and oncologists and surgeons and so on to work on patients with brain tumors. Thank you for your attention.
Wow, really enjoyed that. Thank you so much, Professor. And I really like that quote at the end by Bill Gates. It was very inspiring. But yeah, we just wanted to thank you, especially for you giving your time to us today, with you being on leave. And we also wanted to extend our gratitude on behalf of NANSEG for helping us set up the latest NANSEG neuro-oncology medical center study. We really appreciate that.
Well,
good
luck with it all.
Yeah, thank you. I've had a few questions sent to me directly and some posted on the chat. So I'm going to be asking you a few of those for the next 10 minutes, hopefully. Okay. And I'm also going to post a feedback form in the chat. So if participants can fill that out, that'd be very helpful. And if you fill that out, you'll get a certificate at the end. So that should be good. Okay, so for the first question, how reliable are liquid biopsies, CSF and blood samples regarding sensitivity and specificity, and how often are they currently used in the UK and abroad?
So you'll hear a lot about biopsies in cell-free DNA, microsomes, possibly even tumor-educated platelets. The commonest samples are taken from the blood. CSF, yes, you do get enrichment, but it's much more invasive procedure and doesn't seem to be substantially superior to what you can get out of a blood test. Doing it for brain is different to doing it for epithelial cancers, in that the fragment sizes are smaller and the technology required to identify defining mutations is something that is still evolving. We're in the process of writing up some work from one of the studies that I was involved in Cambridge. It's taken a long time to get this far. There are I have been involved in some work that is published. This is an area that is a work in progress. It is not yet sufficiently evolved to be in routine clinical practice. But again, liquid biopsies, as I mentioned in the talk, is an area that we're very keen to develop, because obviously having access to patients, to the data, to biological tumor samples where we can identify the mutations, and hence would be an infrastructure to validate liquid biopsy techniques. And all of that is part and parcel of what we designed. And taking it forward then is something that we would be very encouraging of people to do. But as I say, at the moment, it's research only, and it's there's still a long way to go for it to become sufficiently specific for diagnostics.
Yeah. Thank you for sharing that. So another question that I got sent to me is, and it goes on to the fact that you talked a lot about, the brain matrix trial and how it can lead to things in the future. So one question was, do you see this replacing existing NICE guidelines in the next 10 years? And if successful, do you think neurosurgeons would be willing to adopt these guidelines straight away?
Will brain matrix be improving? Certainly, we would hope that it will inform the evolution of NICE guidelines and inform policy making. Obviously, if a drug is proved to be effective, then it will automatically be rolled out. I think there's a lot more that can be done around developing and improving on surgical specialization, low volume surgery, and creating an understanding of how surgeons particularly can add value in terms of doing what they're doing, rather than just being the technicians to reduce the tumor for the oncologist. So there's a lot more we can do if we work together.
This is my own personal question that I was thinking of. And so margin sampling is something that I personally haven't come across before. Maybe just because I haven't done my oncology placement yet. And you talked about some of the difficulties it can cause regarding objective sampling and making sure that you sample the right areas. I was wondering if there were any experiences that you could recall where it caused some difficulty in managing the patient due to an inability to objectively sample.
So at the moment, the spatial sampling and the temporal sampling that we've done that have been published are around understanding the biology of the disease and the fact that you might have a drug that targets something that's in a subpopulation of cells, kills all those cells effectively, but the rest of the cells take over and drive the tumor, so you never see a signal of recovery. There have been no examples of how that would work in practice, for the simple reason that there aren't enough drugs around to sort of do that. And that is something that needs to be developed in order to evaluate that. One of the big challenges is you look at all these genomic sequencing and how things have been done to a very high standard, and you can sit there and somewhat cynically say, well, so what? Because it's not made an impact on patients. Glioblastoma is one of the most sequenced tumors. It was one of the first to be sequenced, but it's not really had a big impact. So clearly, it's not just down to the genome. And you've got to remember as well that nature selects for a phenotype, not a genotype. And we don't understand how the genotype translates into the phenotype. That's something we've still got to learn.
Thank you for answering that. So another question that was posted on the chat is there a scope to use single-cell spatial transcriptomics to understand the tissue microenvironment as part of the trial platform?
The short answer is yes. The longer answer is that spatial transcriptomics is where single-cell RNA sequencing is going. It's the next step in refining the whole process, because it's understanding how the mutations are actually translated into something, or I should say transcribed into something, that then influences what happens to the patient, i.e. the clinical phenotype. And that is intimately influenced by the environment in which that is happening. So the immune environment, the metabolic environment, and so on. Even something as simple as how stiff the tissue is. Sometimes when you operate on a patient, the tumor it has a consistency of warm jelly. Other times it's actually quite stiff, like soggy cardboard. And it can be hugely different. But we know that the stiffness the physical stiffness of the environment can influence cell differentiation. So that's just, that's a fourth dimension of pain and complexity to have to deal with.
Another question that just came in. Thanks for a fascinating talk. Do you foresee similar technology and concepts to those utilized in brain matrix being applied to other specialties?
Absolutely. And that's a really interesting question. On one level, you could use this as an example of how we can deal with a rare or an uncommon disease. And any rare or uncommon disease could, in theory, be managed along similar lines. And that's really important because it's understandable that society, that medicine, that funders, that government concentrate on what's common: lung cancer, breast cancer, prostate cancer, and so on. But equally, 50% of patients with cancer do not have one of the common cancers. So by learning how we can deal with the uncommon, by creating networks, that's the way forward.
Thank you. So I had a personal question. I mean, just because of how big and amazing the trial was. So what would you say was the most difficult part of organizing a study of this scale?
Oh, without a doubt, getting people to work together.
Yeah.
It's nothing to do with science. It's all to do with communication, ego, all of that sort of stuff. It's what can be referred to as character building experience, but worth doing.
I'm glad to hear. Well, I think that's all the questions we have so far. We thank you again so much for your time and for the presentation. Really enjoyed it.
Good. And good luck to you all.
Thank you.
All the best.
Thank you. Perfect.
Really appreciate it.
Bye-bye.
Thanks, Prof. You take care. Take care, everyone.